Hey team, Dr. Jake Audie here, and today I'm going to be doing a bit of a review week of the content. I asked you guys what content did you want me to review, and many of you, in fact most of you, said the last 2 weeks. So I'm just going to do a quick summary of the last 2 weeks, and what's great about that is it actually integrates a lot with the previous 10 weeks, and so we're going to get a good overview of the course. Okay, I'm going to try to do the whole course. Sure, why not? Now, a general little pro tip from a lecturer is that whenever you are in these review weeks or review tutorials or review lecturers and you hear the lecturer say, this is really important, or I would focus on this, they're giving you a good hint about what might be in the exam. So make sure you listen to your lecturers, figure out what they get excited about, what are they telling you is super important, and it might tell you where some of those trickier questions might lie in the exam. So I'm going to start with antibody responses up here, and here we have a nice picture of a secondary lymphoid organ, right? So what's a secondary lymphoid organ? Well, a good start is what's the primary? A primary is where the adaptive immune cells are produced and matured, right? So this is your bone marrow, because your B cells are produced and matured there, and this is your thymus. These are the primary ones, because your T cells are produced by your bone marrow, but they go to your thymus to mature. A secondary lymph organ is where mature immune cells sit and wait for a pathogen or an antigen or a pathogen-associated molecular pattern in order to respond to it. So they scan the lymphoid tissue, the lymph fluid, for example, or the blood. Now, 2 good examples is a lymph node, which is scanning your lymph fluid, and your spleen, which is scanning your blood. Now, I believe this is a lymph node right here, although at this zoom it is kind of hard to know because they have similar structures and when it comes to adaptive immune cells. But so in red we have B cells, in blue we have T cells, and in green we have proliferating cells, Ki67. And what we can see here is a red circle here. This is what we'd call the follicle and of the lymph node, and this is where B cells sit. Now we can see in an extrafollicular space is where the T cells sit, and there is a border between them, right? And this is where the B cells and T cells are going to interact. And then in the middle, we've got a bunch of proliferating cells. In the middle of the follicle, we've got a bunch of proliferating cells, and it's where a high density B cells are and T cells are. Now, these T cells are follicular T helper cells, or T follicular helper cells, and the shorthand for that is TFH, and that's because they're in the follicle. That's why they're called follicular T helper cells. And where the follicular T helper cells are and the B cells are, we get this massive proliferation. And that's because this is called the germinal center of the follicle, and it's where the B cells are dividing, class switching, and undergoing selection for the highest affinity antibody producing B cell we can find. So it's sort of like the evolutionary survival of the fittest center of the lymph node, because the follicular T helper cell is going to be helping to select the B cell with the best antibodies, and then the B cells are going to mutate, and then the follicular T helper cell is going to check, is that even better, and so on and so forth, right? So that's why there's so much division going on in the germinal center. But some B cells will just go straight away and get activated, and they'll become extrafollicular B cells, also known as extrafollicular plasma cells. So at that border, the B cell's got a decision. Does he go extrafollicular, or does he go into the germinal center to get, survival of the fittest and become the best B cell he can be? Right. So, lymphoid organs, primary, as I said, bone marrow, thymus, secondary, tonsils, but also lymph nodes, spleens, that kind of thing. And here we can see that one thing I just missed out on my previous discussion was where are the APCs? Now, the APCs are floating around here, and here, this is the spleen, that's the lymph node, and you can see they've got that similar structure of green B cells, T cells, and we've got APCs. So the APCs will activate the T cells will go to the T cell B cell border, they'll interact, and they'll tell the B cell what to do. Should you go become an extrafollicular B cell, producing low affinity antibodies, or should you go into the germinal center and become the best B cell you can be and class switch from an IgM antibody into the IgG or IgA or IgE antibodies? All right, so all of what I've been talking about is what we call a T-dependent antibody response. And that's because it's dependent on the thymus. A lot of the experiments were done where they would remove the thymus. So therefore you have no mature T cells in mice. And so they could distinguish between B cell responses that were dependent on the thymus and thus the T cell. And so they became T-dependent antibody responses. So this is the classic T-dependent antibody response. You've got activation of the B cell receptor. It's now taken in that pathogen and is expressing the an antigen on its MHC2 molecule following digestion and then presentation, and this has activated a T helper cell at that T cell B cell border, and it is now releasing cytokines to tell that B cell what to do. So it requires that 3 steps: BCR activation, TCR activation, and cytokine production. And we get basically the division, and either they divide and become memory cells. And they're memory because, remember, each B cell has its own unique antibody gene, which it has gone through a somatic recombination, which has mean out of about 40 gene segments, it's removed most of them. It removes like 35 of them, 33 of them, and leaves itself with only 5 or 7 of these variable fragments left in its one gene. So the gene used to have 50 segments, and now it's been cut down. And so now it has that memory of what its antibody looks like because it's genetically engineered itself. So the memory is encoded in its DNA. That is where the memory is, in the antibody gene. Or it can go on to become an effector cell, aka a plasma cell. A lot of clinicians and doctors will use the term plasma cell, and that's because they've got all this rough ER in there, which increases its plasma, its cytoplasm, right? There's a lot more of that in there. And that's so it can pump out loads and loads of antibodies. Now, this is a new thing introduced by Gunnar. It's fantastic. It kind of explains what IgM is. So this is a T-independent antibody responses. So scientists noticed if you take the thymus out of mice, you can still get antibody responses. And so they're thymus independent or T cell independent antibody responses. And this is because B cells have pattern recognition receptors, PRRs, that can recognize pathogen-associated molecular patterns, PAMPs, like LPS or bacterial DNA. So once you get that pattern recognition, like a TLR, for example, or toll-like receptor, once it recognizes a PAMP and you've got an antigen present on the B cell receptor, this guy is going to activate proliferate and start to produce antibodies. What antibody? IgM. It's going to produce IgM antibody in a T-independent response. And this is why, remember this slide, I went through really briefly all the different subtypes of antibodies. IgM is the early antibody. And one of the reasons of this is that it doesn't require T cells for you to start to pump out IgM. So that's one of the reasons why it's the early antibody. But IgM isn't that great. That's why IgG is the good antibody and IgM is the early antibody. So IgM has a short lifespan. It has relatively low affinity. And what I'm about to cover is going to explain why it has low affinity. And it's mostly about complement activation. So it's not great at some of the other things like neutralization. And that's because it has a low affinity and a short lifespan. So it's actually not good at some of those responses, but it's very good at complement activation. It can agglutinate and precipitate, but again, because it's low affinity and short lifespan, it will break down and then release the pathogen. And because it doesn't have a really tight bond on it, it's not amazing at those processes. It's mostly about complement activation. So how do you get the other antibodies, right? We've got IgM, but we want other antibodies. So we're going to go through what's called a process of class switching. So we're going to switch away from IgM to the other antibodies. And how do you do this? Well, this is where, so we got to that next level of complexity about this, right? And so this is where the T helper cells come in, right? So we've got Th1, Th2, and Treg. Now, when I presented it, I said IL-4 causes the antibody production, right? But in truth be told, other cytokines are critical too. In fact, there's loads and loads of other cytokines that are critical too, and they can help with choosing what class. So IL-4 will actually help produce IgG, IgE, and IgA. So this is why I chose IL-4, is it is it causes the production of antibodies, and it also stimulates the extra production of antibodies. It's most famous for IgE in that Th2 response, but IL-4 also helps with the other antibody responses. But interferon gamma alone can also help with, preferentially cause the IgG production, right? And that makes sense, right? Because when we're talking about IgE, we're talking about that histamine response, right? We're talking about killing off a worm with histamine and activating mast cells and basophils and eosinophils. And remember, eosinophils are full of neurotoxins, right? So none of those processes, histamine, eosinophils, and all that, can help with viruses, right? So IgE isn't great for viruses. So what we need is the combination here to get the IgG, which is high affinity, long-lasting, fantastic in neutralization, fantastic in neutralization and activating Fc receptors on macrophages to cause phagocytosis, and it activates, we're about to go through this, Fc receptors on natural killer cells, which can help with killing cells. So IgG is the good antibody. It does so much stuff. And IgA is about being secreted. So IL-4 does help with this, but so does TGF-beta. And you might go, why is TGF-beta good for IgA? Well, remember, IgA, it goes out your anus because it's secreted. It also secreted in your tears and in the breast milk, but it is a secreted antibody. So once you start to clear what's in you, you want to start to secrete it to protect what's out of you, what's in your intestine and what's in your nose and that kind of thing. So that's sort of how I think about it when it comes to the resolution process. We've got a Th2 response, which is going to produce and we've got a Treg response, so we're trying to calm down our immune system. We've cleaned up what's on the inside. Now we want to keep secreting stuff on the outside and maybe prevent reinfection and that kind of thing. Right, this is really important, guys. Really important. The fate of the B cell. So here is the lymph node. We've got the germinal center right there in the middle of the follicle. The follicle's this thing right around here. So the follicle are where B cells are, and the germinal center is where that maturation, proliferation, and that selection process goes on. Here is the T cell, B cell border, and the interfollicular area where the activated T helper cells are, and the paracortex is where naive T helper cells meet APC. So this is where a dendritic cell presents to a T cell, it gets activated, it goes to here, it goes to the border here where the B cell and the T cell start to interact and talk to each other, and then we can get this response. Now, this is key. This is super key. There are two fates of the B cell, right? The B cell, once activated by a T helper cell, the B cell can become an extrafollicular plasma cell. And this happens super quickly, right? So there's that interaction between the T helper cell and the B cell. We get that T cell receptor interaction. We get the cytokine release. We get the communication. And then we get a quick division of that B cell. And it will start pumping out IgM as well, right? So this is a very fast response. It is T cell dependent, very quick. And we get that IgM response. And it is... IgM, remember, is short-lived and low affinity, so that's not great. So we get an extrafollicular plasma cell, and that happens very quickly. What can also happen, this is the two fates of the B cell, very important, is the germinal center. So it can now migrate into the germinal center, which is here. So it's migrated from out here into here. Remember that bright green spot in the immunohistochemistry image, and here it's going to interact with that T follicular helper cell. And so we've got a germinal center B cell, not an extrafollicular plasma cell, and it's interacting with a T follicular helper cell, and this is where it's going to undergo class switching and this evolution to produce high affinity antibodies. So how does that work? Well, how do we get a T follicular helper cell? Just quickly, super briefly, you need IL-21 and IL-6 Typically, IL-6 is a general inflammatory cytokine produced by antigen-presenting cells in response to PAMPs. Really quickly, and you get IL-21. And then IL-21 is what helps the sort of maturation process. So if a B cell produces a not very good antibody that doesn't bind to the antigen very strongly, through the immune synapse between the T helper cell and the B cell, we can make that, not immune synapse, but through that cell-to-cell interaction between the T helper cell, T follicular helper cell, and that B cell, we can get that selection process. It will either produce IL-21 if it's a high affinity antibody, or if it's not a high affinity antibody, the follicular T helper cell won't produce IL-21. And so we get this selection process where it goes round and round and round, and it will start to class switch into IgG typically, but also the other antibodies depending on what other cytokines are around. But typically IgG, and that IgG is going to get higher and higher affinity as the good B cells that have undergone a mutation that has made that antibody even stronger goes round and round and round, gets selected by the follicular T helper cell. If the follicular T helper cell says, no, I don't like that, it's gonna, the B cell's gonna undergo no division and cell death. So it's sort of a selection process to produce stronger and stronger affinity antibodies. But the key thing here is these two fates here, and what cells are driving those two fates here. So we end up with this sort of interaction here and the class switching and the evolution to higher affinities. So up next in the next video, I'm going to go over cell killers.